In this series of videos, I'll be covering the histology of the pituitary gland. Remember that endocrine glands secrete their products into the surrounding connective tissue stroma, rather than into a duct like exocrine glands. You can see them here, where you have the collections of the endocrine cells secreting into their surrounding matrix. From there, those secretions, which are hormones, enter the vasculature. So typically endocrine glands have a rich vascular network. Via this vasculature, those hormones reach their target tissues or organs. The major endocrine organs are shown in this diagram here at the right. And you can see pineal and pituitary up in the cranium, as well as the thyroid and parathyroid here in the neck, the adrenal and the pancreas, and of course the gonads are included too. I'm not sure why the thymus is even on this diagram because it's an organ of the immune system, but perhaps it's just to contrast it so you don't confuse it with the thyroid, which is also in a similar location. So the pituitary gland, also called the hypophysis, is a very small pea-sized oval organ that is suspended from the underside of the brain by the pituitary stalk, which in turn is connected to the hypothalamus. You can see down here in this diagram, there's the pituitary and there's the stalk, and this is the hypothalamus in this area right here. The pituitary is located within the sella turica, which is a small depression in the sphenoid bone. So many of the other endocrine organs that were shown in that original diagram a couple of slides back receive input in the form of stimulating hormones from the pituitary gland. Therefore, the pituitary is sometimes called the master gland. It is attached to the hypothalamus, shown in this diagram at a little bit higher mag than the previous slide. And the hypothalamus, in turn, contains neurosecretory cells and controls the release of the pituitary hormones. So let's start by looking at the structure of the pituitary. It is essentially a compound endocrine gland, which consists of two lobes that are very disparate from one another. The first is the anterior lobe, called the adenohypophysis, which is comprised of epithelioid glandular tissue. Like most glands, this contrasts with the posterior lobe, called the neurohypophysis, which is really neural secretory tissue, and it looks like other nervous tissue that you have studied in the past. Let's start with the posterior lobe. It has two portions. First of all, the stalk that comes from and connects to the hypothalamus is called the infundibulum, and that is shown right here, coming down from this median eminence of the hypothalamus. And the main part of the posterior lobe, which is called the pars nervosa. The anterior lobe has three parts. The main part of the anterior lobe is called the pars distalis. There's a small portion called the pars intermedia that is around this series of cystoid spaces. And then there is a portion that reaches up and kind of wraps around the infundibulum called the pars tuberalis. So why is the pituitary comprised of two such disparate regions? The reason is, embryologically, it's derived from two different sources. The posterior lobe is derived from a downgrowth of the neuroectoderm, and that's shown here, the beginning of that downgrowth. The anterior lobe is derived from an upgrowth of the oropharynx, and that's shown here. During this upgrowth, this oropharynx folds on itself and forms something called Rathke's pouch. As this process continues, it pinches off and you get the remnant of Rathke's pouch, sometimes called Rathke's cysts, which is that series, eventually becomes that series of cystoid spaces that I showed in the mature tissue. The oropharynx loses its connection to this portion of the pituitary that's going to become the anterior lobe, whereas the posterior lobe portion retains its connection, and you can see that here. Let's talk for a minute about the blood supply of the pituitary and the hypothalamus. So the superior hypophyseal artery here supplies the pars tuberalis, the infundibulum, and the median eminence of the hypothalamus. And you can see the capillary network here in this area. This capillary network in turn drains into the portal veins, the hypophyseal portal veins shown here in purple. And the portal veins give rise to a second capillary network in the pars distalis. That means the neuroendocrine secretions of the hypothalamus in the median eminence are released pretty directly right into the pars distalis, and they stimulate the production and secretion of the anterior lobe cells. Now, the inferior hypophyseal artery supplies the pars nervosa here, and is only very peripherally connected between the two lobes. So what this really means is that the pars distalis does not have a direct arterial supply. It's only supplied via the portal veins. So let's look at the structure of the anterior lobe and the function of the cells that are there. Essentially, the anterior lobe of the pituitary regulates other endocrine glands and some non-endocrine tissues as well. In the pars distalis, the cells are organized into clusters and cords, which are invested by the fenestrated capillary network surrounding them. Histologically, there are three types of cells that you can identify, and their identification is based on whether they stain in an acetophilic, basophilic, or not at all fashion. So those that are staining bright red to pink are called the acetophils, those that are staining the dark purple are the basophils, and those that are very pale staining and lack either one of those other staining characteristics are going to be called chromophobes. What are these cells specifically, and what hormones do they make? So there are two different cell types that stain in an acetophilic fashion. Those are somatotropes and lactotropes. Somatotropes make growth hormone, and they are the majority of the cells, up to about 50% of the cells in the pars distalis. The lactotropes make prolactin, and they are between 15 to 20% of the total cells there. Then there are three types of cells that stain in a basophilic fashion. Those are the corticotropes, gonadotropes, and the thyrotropes. Corticotropes make adrenocorticotropic hormone, and they also have about 15 to 20% of the total cells there. Gonadotropes make two hormones, follicle-stimulating hormone and luteinizing hormone, and those gonadotropes are about 10% of the total cells. And the third basophilic cell type is a thyrotrope, and that makes thyroid-stimulating hormone, and those are about 5% of the total. The chromophobes are probably quiescent or stem cells. It's not really known exactly what they're doing, and they don't make hormones. They could be any of the others, acetophils or basophils, in a resting stage or in a stem cell stage, and that's not been determined. So I said previously that the hypothalamus makes neurohormones that control the anterior lobe hormone release. So how does that work? So the release of growth hormone from the somatotropes is regulated by growth hormone releasing hormone from the hypothalamus. It's also inhibited by somatostatin, which is made in the pancreas, and by enteroendocrine cells in the pyloric stomach and duodenum. Ghrelin, which is made in the stomach, also stimulates the release of growth hormone. The release of prolactin by lactotropes is regulated in several ways. First, by dopamine, which inhibits its release, and that's made in the hypothalamus. Second, by thyrotropin releasing hormone, also made in the hypothalamus, and by vasoactive inhibitory peptide, which stimulates the release, and that's made in the hypothalamus, the gut, and pancreas. ACTH release is regulated by corticotropin releasing hormone from the hypothalamus, which stimulates the release of it, and follicle stimulating hormone and luteinizing hormone made by those gonadotropes are regulated by gonadotropin releasing hormone from the hypothalamus, which stimulates their release. Finally, TSH, or thyroid stimulating hormone, made by the thyrotropes, is regulated by thyrotropin releasing hormone, which stimulates its release, and that's from the hypothalamus, and somatostatin, which inhibits its release, and again, that's made in the pancreas, the pyloric stomach, and duodenum. Now let's look at the other areas of the anterior lobe, the pars intermedia. So this is the portion of the anterior lobe surrounding those cystic cavities, which are sometimes called Rathke's cysts, and we'll probably call them those when we look at them in lab. They're really the remnant of Rathke's pouch from the embryological developmental stage. This area is populated by basophils, and those are mostly corticotrophs, and there's a lot of chromophobes here as well. And you can see I've kind of outlined this area, and it's squeezed between the main area of the pars distalis and the pars nervosa. And the reason that this area is distinct from the pars distalis at all is that when these two lobes are dissected, the pars intermedia tends to stick to the pars nervosa, which is kind of opposite of what you might expect, but that's the way it works. The final area, called the pars tuberalis, is an extension of the anterior lobe coming off of the pars distalis and wraps around that infundibulum. And you can see the pars distalis here, and the pars tuberalis kind of going up around this infundibulum here. So the pars tuberalis is a highly vascular region, and the parenchymal cells are arranged in small clusters or cords in association with the vasculature there. These cells often show immunoreactivity for ACTH, FSH, and LH, which means they're corticotropes and gonadotropes.